Hello, everyone, and welcome to our lecture number 2 of Carbohydrate Metabolism series. Having completed the foundational lectures on the chemistry of the carbohydrates uh, from the lecture number 1 to 6, and after the digestion and absorption of the carbohydrates in lecture number 1 of the Carbohydrate Metabolism series, now we move inside the cell to explore the utilization of the glucose molecules. This lecture is clinically very important because every single organ in the body depends on the precise regulation of blood glucose levels, and glycolysis is the central pathway to produce the energy that converts the glucose into energy, either both aerobic and in anaerobic conditions. Right? Disruption under hypoglycemia, diabetes complication, lactic acidosis, hemolytic anemia, and even the Warnberg effect in the cancer, right? So let's proceed step by step with the clear biochemical mechanisms and their direct clinical correlation. So for our ease, I'll divide this very lecture into two parts. The very first part would be the utilization of glucose, and the second part would be regarding the glycolysis and the stages of glycolysis, right? So let's start with the withdrawal of glucose from blood. So once glucose enters the bloodstream, after absorption, the blood must either store or use it immediately. The two major withdrawal processes are, the very first one is the glycogenesis, that is the conversion of glucose into glycogen that primarily occurs in liver and skeletal muscles. The second one is the conversion of glucose to other compounds, including fats, that is done by the lipogenesis, and other carbohydrates or amino acids, right? So clinically, insulin is the key regulatory hormone, drives these processes. In insulin deficiency, like in case of type 1 diabetes and insulin resistance, as one can see in type 2 diabetes, withdrawal of glucose from blood is impaired, leading to hyperglycemia. And its long-term complication like neuropathy, retinopathy, and nephropathy can occur in these conditions. So insulin is the key hormone driving both these processes, the glycogenesis and the conversion of molecules or the glucose molecule to the other compounds, right? Next is the release of glucose by the liver to the blood. The liver maintains liver or the blood glucose during fasting by two opposing pathways. The very first one is the glycogenolysis, that is the breakdown of the stored glycogen to free glucose level, right? The second one is the gluconeogenesis. Gluco means glucose, neo means new, genesis is the production, right? So the synthesis of new glucose from the non-carbohydrate precursor molecules, that those are the lactate, the glycerol, and the amino acids, right? So these processes are stimulated by glucagon hormone and epinephrine and inhibited by insulin. So clinical correlation at this very point is this: in glycogen storage disorders, for example, in von Gilkey's disease, the glucose 6-phosphatase deficiency occurs. Glycogenolysis and gluconeogenesis fail in these conditions as well, causing severe fasting hypoglycemia, hepatomegaly, and lactic acidosis. So what are the factors determining the blood glucose levels, right? So let's discuss all these in detail. Blood glucose concentration at any moment is the net result of glucose withdrawal that is being done by glycogenesis and the utilization of glucose by the body versus glucose release that is being done by the glycogenolysis and gluconeogenesis processes by the liver. The functional integrity of the liver is very critical in order to carry out both these processes, either in case of to produce energy or to release energy that are being stored in the liver, right? So liver failure in case of cirrhosis, acute hepatitis impairs both storage and release, leading to unpredictable glucose swings. And in critical care or ICU setups, this slide reminds us why we monitor blood glucose hourly in hepatic encephalopathy or in shock-like conditions, right? So, what are the major fates of glucose? Glucose has several metabolic fates, like the oxidation. Oxidation via glycolysis, right? That is energy yielding pathway to pyruvate or lactate, right? HMP shunt, pentose phosphate pathway generates NADPH molecule and pentoses, and there will be no net ATP produced during this process, right? The uronic acid pathway, in case of mucopolysaccharides and glucuronide synthesis, right? The next fate or the metabolic fates would include the storage as glycogen in liver and skeletal muscles, conversion to fats, conversion to other carbohydrate molecules, and the conversion of the carbohydrates to non-essential amino acids. So this overview is essential because it shows how glucose metabolism integrates with the lipid, nucleotide, and protein pathways, a concept tested heavily in exams, every single exam, and relevant to obesity, diabetes, and inborn errors of metabolism, right? So this was all about the utilization of glucose, right? So you have now mastered the utilization of glucose by discussing some of the processes or the major phase of the glucose, along with by having the release of glucose by the liver to the blood and the withdrawal of glucose from the blood in case of glucose, a single molecule that is pretty important for every single activity of your life. So thank you very much for watching this video. And the second part, I'll continue with the glycolysis, that is the production of pyruvate or the lactate from the glucose or the glycogen. And we will divide this pretty step or the process into 4 stages, and we'll discuss in detail the types of glycolysis, aerobic and anaerobic, and definitely you're going to learn a lot. Thank you once again for watching this video.